Hello everyone. In this presentation, we are going to see the histology of liver and we will try to correlate how the tissues in this organ are organized to perform its function. As we all know that the liver is the largest organ of the body and it performs several of the important functions such as synthesis of urea, bile, detoxification of several metabolic wastes and drugs, gluconeogenesis, etc. Before going into the details of the microscopic structure, let us see few details of gross anatomy of the liver. So this figure shows the anterior surface of the liver and you can see it is divided by falciform ligament into right and left lobes. And in this figure we can see the posterior surface of the liver, which is related to several of the visceral organs, and we can also see caudate lobe, quadrate lobe, and in between the hilum or the porta hepatis. Porta hepatis is the region through which structures enter and leave the liver. The structures entering is the hepatic artery, the portal vein and plexus of nerves. The structures leaving are the hepatic ducts and some lymphatics. Let us see the microscopic features of the liver. Like any other organ, histologically, liver can also be subdivided into two components, the stroma and parenchyma. The stromal part, which is responsible for providing the structural support to the parenchymal tissue. Stroma is the connective tissue capsule, which is also known as Glissen's capsule, which covers the entire organ. And at the porta hepatis, this connective tissue capsule is going to enter the liver, and inside it is going to subdivide the parenchyma into several structural units. As this connective tissue enters the hilum, it branches like a branch of tree and it keeps on dividing the parenchyma into several structural units which are known as classical hepatic lobule, and we will see the details of this in subsequent slides. The parenchyma is consisting of the cells which perform its function. It is that component of the organ. And the cells, the way they are organized, they have been described in three ways. One is the classical lobule, which I was discussing along with the stroma, how it is organized. And there are other two ways to describe the parenchyma. The portal lobule, this is also a structural unit, and then a functional unit, the hepatic acinus. The classical lobule is also known as the hepatic lobule. This is one of the ways to describe, one of the three ways to describe the parenchyma. So this parenchymal unit is surrounded by the connective tissue. As described earlier, the connective tissue enters the porta hepatis and subdivides the parenchyma into this units. These are called as hepatic lobule or the classical lobule. Each lobule is hexagonal in shape. Having 6 angles, and in the center of the hepatic lobule is the central vein, and at the 6 angles of this hexagonal unit there are 3 tubular structures. These are together called as portal triads or the portal areas. The largest lumen of these three tubes is of portal vein. So that's the portal vein. Portal vein. Then there is hepatic artery. And the third tubular structure is the bile duct. And occasionally there will be another tubular structure, the lymphatic. So that's the bile duct. This is the having the smallest lumen among all the three tubular structures. So these three together constitute portal triad. And the main parenchymal cell is the hepatocyte, which is polygonal in shape, having a centrally placed round nucleus. This hepatocyte is arranged in this hexagonal unit. Within this hexagonal unit, as single or double celled plates. Now, what I mean by the plates is, when you see it in 3 dimension, this figure which I have drawn is in 2 dimension. So if we see these organization of these cells in 3 dimension, it can be compared with the wall of a brick. So if we see the wall of a brick, so these are the bricks. So that's the organization of the wall of the brick. Similar organization is there for the hepatocyte. Now you have to consider this wall is made up of 2 bricks. So the wall is made up of 2 bricks. Similarly, the hepatocytes are also arranged in plates like this, and they can be single celled or double celled as shown here. It will be more clear when you see the next slide. So that is 1, 2. There are 2 classical lobules here. In the center is the central vein, and surrounding the central vein, all these are plates of hepatocytes. They are arranged so that they appear radiating from the central vein. These plates, as I mentioned earlier, are 1 or 2 celled thick, and you can see here the hexagonal unit, the parenchymal, the classical lobule, parenchymal tissue are organized in a hexagonal structure known as the hepatic lobule or the classical lobule, and on each angle it has 3 tubular structures, the portal triad. Let us see in another schematic figure. these are hepatocytes. These represents the hepatocytes, 1 and 2 cell thick. These plates of hepatocytes are separated from neighboring hepatocytes by hepatic sinusoids. We will see this hepatic sinusoids in next slide, details of hepatic sinusoids. These plates of the hepatocytes, they may anastomose with each other. And in this figure, you can also see this is the portal vein. So the blood from the portal vein goes into the hepatic sinusoids. At the same time, that's the hepatic artery, and the blood from the hepatic artery also enters the hepatic sinusoids. And then this blood enters the central vein. That's the central vein in the center of the classical lobule. You can also see the bile duct here. We will talk about the blood flow and flow of the bile in subsequent slides. This is an hand-drawn figure of classical lobule, classical hepatic lobule in a low magnification as it is seen. So you can see here, these are the hexagonal units, the hepatic lobule. In the center is the central vein, and on the angles you can see the portal triads. That's in the hand-drawn picture of magnified view. So you can see the plates of hepatocytes, there you can see the portal triad, there you can see the central vein, and you can see some amount of connective tissue separating these units. This is photomicrograph of the liver showing the classical lobule in low magnification. So in a human tissue, the hexagonal shape is not very clearly defined, whereas in compared to the animal tissue where it is very clearly defined. So here we try to see the central vein, and then we try to locate the 6 angles. So that's the central vein in the center, and that's the portal area, the portal triad. There again the portal triad. You can see the 3 lumens. I'll show in the next slide in the magnified view. So this is one lobule. That's the magnified view. You can see here the hepatocytes, that's the central vein, and that is the hepatic artery, and that is the bile duct. And in between the plates of the hepatocytes, this is hepatic sinusoid. In this slide, we will see the details of hepatic sinusoids in space of DC. The hepatic sinusoids receives the blood from the hepatic artery and portal vein, and it conveys this blood to the central vein as described in the classical lobule. These hepatic sinusoids are fenestrated capillaries. So the cell lining these are the endothelial cells lining the capillaries, and there are another cells. These are Kupffer cells, the phagocytic cells, and these are the endothelial cells. I am drawing the endothelial cells on the other side. That's one more phagocytic cell, the Kupffer cell, and sinusoids, the endothelial cells in the sinusoids. And the blood flows here. I am trying to show the RBCs present in the hepatic sinusoids. So these are the fenestrated capillaries with two types of cells in their wall, and on either side will be the hepatocytes. So that's the hepatocytes, and the surface of the hepatocyte facing the sinusoid has microvilli to increase the surface area for exchange of material. So there will be the hepatocytes here. I will draw one more on this side. That's the hepatocyte. And the space between the hepatic sinusoid and the hepatocyte, this space here is the space of Deci. This space is space of Deci, and it is this space where the exchange of substances between the blood and hepatocytes occur through this space. There is another cell present here. These are the lobules in that. That's hepatocytes. So this cell here is known as etho cell, or it is also known as hepatic stellate cell. They store vitamin A, and they may perform some other functions. That's almost the same picture which I have drawn. So here is the hepatocyte with its surface facing the space of Deci, showing the microvilli there. These are the endothelial cells. That's the Kupffer cell. That's the RBC in the lumen, this one. and this space is the space of Deci. That's the etho cell or the hepatic stellate cell. One of the ways to subdivide the parenchymal units is based on the flow of bile, and these units are known as portal lobules. So before going into the details of portal lobule, let us see the flow of bile. So the hepatocytes will secrete the bile into small ducts which are lined by the walls of the hepatocytes. So like that. So these are the hepatocytes here. These are the hepatocytes. So these are bile canaliculi. These are lined by the hepatocytes. And they are joined, they are separated from the intercellular space as they have occluding junctions. So the lumen of the bile canaliculi is separated from the intercellular space. Now as these canaliculi unite, then they have their own cells, cuboidal cells in their wall. One layer of cuboidal cells. So and so on, like that. These are bile ductules, or they are also known as canal of herrings. And several of these bile ductules unite to form a bile duct which is present in the portal triad, and these ducts will unites together to form another larger duct, and eventually they all form the hepatic duct. So this is how the bile flows. So it flows in the opposite direction to the blood. Now, as I mentioned earlier, the portal lobule is based on the flow of bile. So this is another way of subdividing the parenchyma. So this is triangular shaped parenchymal units, and at the three angles of this triangular shaped unit is the central vein. So this is one central vein, then is another central vein, and that's the third central vein of the three neighboring classical hepatic lobule. And in this portal lobule, in the center is the portal triad. So the bile secreted by the hepatocytes in this triangular region is collected into the bile duct present in the center of this triangular unit in the portal triad at this location. So all this bile here will come to the bile duct present in the portal triad, which is present in the center of this triangular unit. So that's the portal lobule. Hepatic acinus is another way by which the parenchyma of liver can be subdivided into parenchymal units, and this is based on the blood supply of the liver. So before going into the hepatic acinus, let us see the blood flow within the liver. So the blood to the liver comes from two sources: the hepatic artery, this contributes to around 20%, and portal vein, this constitutes around 80% of the blood to the liver. And the blood which is brought to the liver by portal vein is rich in nutrients, as it is coming from small intestine, and also in breakdown products of hemoglobin, as it is coming from spleen. So these two vessels, the hepatic artery and portal vein, will enter the liver through the porta hepatis along with the connective tissue. And as I explained earlier in the classical lobule, that these vessels accompany the connective tissue, and in the classical lobule they are present at the 6 angles of the hexagonal unit. I am drawing only one. There are one hepatic artery and one portal vein, and the blood from these will be received by central vein. That's the central vein. So the blood from these two vessels will be received by the central vein through hepatic sinusoids. So let's see the blood vessels with this diagram. This represents the blood flow in a classical lobule. So there are these 6 angles, the portal triad at 6 angles, and each having a portal vein, hepatic artery, and the blood from the artery will enter the hepatic sinusoids. Also the blood from the vein will enter the hepatic sinusoids. So the sinusoid has arterial as well as venous blood, and the blood is drained into central vein here, and this, in this figure, this part has been shown. I have tried to show the interior of the central vein where numerous sinusoids are opening. These are openings of the sinusoid. So the central vein drains into a larger sublobular vein, and eventually several sublobular veins will unite to form the hepatic vein. What is hepatic acinus? It's a diamond shaped region in the parenchyma. So this unit has 4 angles, a pair of opposite angles. So at 2 opposite angles there is central vein. So at these 2 angles there are central veins, and the other 2 opposite angles, at this point there are portal triads. 2 opposite angles, there are portal triads, and this area, diamond shaped area, is the hepatic acinus. Now, as you know, the blood vessels present at the the blood vessels present at the portal area, the portal triads, they will give the branch in the connective tissue. So they give a branch, and branches from these vessels will deliver the blood into the sinusoids. And the hepatocytes in this entire region of the acinus will receive its supply from this vessel here. So obviously the hepatocytes close to this blood vessel will have more access to the oxygen present in the blood compared to the hepatocytes which are little away from the blood vessel. So based on the delivery of oxygen and the nutrients, the three regions have been defined in the acinus. Three zones. So that's the first zone. That's the second zone. And that's the third zone. So the first zone has best oxygen supply and nutrients. Next comes the second zone, which is less than the first zone, which receives lesser supply than the first zone, and the least is the third zone, which is the farthest from the blood vessel. And hence in disease process the hypoxic changes occur first in zone 3 in this region because it is farthest from the blood vessel. So that's the importance of the hepatic acinus. Thanks for listening to me, and I hope that you will find this presentation useful. Thank you.
Buy the book now at your nearest bookseller or online.